the local sequence alignment through dynamic programming method. As the name indicates, herein query sequence is relatively smaller as compared to your test or reference sequences. So local sequence alignment is carried out or it is processed for sequences which are of different length. In addition, they are also distantly related. Herein we find local regions of alignment within the target sequence. So in most of the cases, this local sequence alignment is used to identify conserved regions within the protein sequences or the nucleotide sequences. There are certain proteins or genes which perform similar functions in different kind of or distantly related organisms as well, like the cytochrome P450 gene. It is involved in like photosynthesis or energy production. So it is a highly conserved sequence. Further, this local sequence alignment is also useful for identifying cis-regulatory elements within the promoter sequence. What are promoter sequences? Promoter sequences are present upstream of the site where the transcription factors are going to bind or transcription process is going to initiate. Process of transcription results in the formation of mRNA. So if this mRNA synthesis is going to start from here, these promoter sites on the upstream of this, there are certain regulatory elements are present which are highly conserved, and these sequences or these elements are called as cis-regulatory elements. So this local sequence alignment is useful for identifying these six regulatory elements. Moving forward, now we will see how local sequence alignment can be carried out using the Smith-Waterman algorithm. Like the Needleman-Wunsch algorithm discussed in my previous video, for every match between the pair of nucleotides, a score of +1 is applied. For every mismatch, a score of -1 is applied. Like for example, A does not match with C, or A does not match with T, or A does not match with G. In addition, a gap penalty results in a score of -2. However, all negative values are considered to be 0. This is a very unique point of Smith-Waterman algorithm. Unlike the Needleman-Wunsch algorithm, which does not consider the negative values to be 0, the Smith-Waterman algorithm specifically narrates or specifically it points out that all negative values must be taken as 0. For filling the values in the matrix square or the box, we first calculate the following values. The score in the subsequent box is based upon these three scores. That is, what is the score in the upper box, what is the score in the left box, and what is the score in the diagonal box. So to the upper box score, a gap penalty of -2 is added. Likewise, for a left box score, a gap penalty of -2 is added, and for a diagonal box score, a score based upon match or mismatch is provided in the subsequent box. So the maximum score acquired after these three steps of scoring is added here. We'll see in the very next slide how this calculation is performed. So herein we have taken a two stretch of nucleotides. This stretch of nucleotides reads as A, T, G, C, G, C and A. With this stretch of nucleotides is placed horizontally above the matrix, and the other stretch of nucleotides is placed vertically downwards, and it reads as A, C, G, T and A. So the very first stretch of box here is taken as 0, unlike the Needleman-Wunsch algorithm where the first stretch of boxes or the first line of boxes both horizontally and vertically, they were filled with negative values. Since Smith-Waterman algorithm considers all negative values to be taken as 0, so that is why the negative values have been replaced with 0 in this case. So now we talk about what should be the value in this particular box. For this, we'll first check what is the score between the previous box which is placed horizontally. So here in the score in the previous box was taken as 0. We will add a value of gap penalty of -2 here. So a value of 0 will be added with the gap penalty, that is -2, and it will result in 0 minus 2, that will be equal to -2. Fine. Now we'll see what is the value in the vertically placed score above the box which is to be tested or which is to be filled. For the box to be filled according to the score present in the previous box placed vertically, we will add a gap penalty of -2. The score in the previous box was 0, as you can see here on your screen. Again we will add a value of -2. So 0 plus -2 will be equal to 0 minus 2. That will be equal to -2. Next, we check what is the score in the previous diagonal box. Herein, the previous diagonal box does not have any score. It is taken as blank, so it means the score is 0. We will then check whether there is an alignment between the set of nucleotides. Of course, A is matching with A, so it means there will be a value of +1 between the stretch of nucleotides. We'll add the value of +1 here. Now we'll move to the next box and check what is to be filled here. Now this box to be filled is labeled as purple. So the score in the previously horizontal box was 1. We'll add a gap penalty of -2. 1 plus -2 will be equals to 1 minus 2, and this will result in a score of -1, and the score of -1 will be filled in this box temporarily. We'll again check according to the score present in the vertically placed box. So herein the score in the vertically placed box was 0. Gap penalty is -2. So 0 minus 2 equals to -2, and a value of -2 is placed in the box. Now we'll see what happens when we move along the diagonal direction. So herein A does not matches with T, so it becomes very clear that there is a mismatch, and we will add a mismatch score of -1. And the score in the previous diagonally arranged box was 0. So 0 plus -1, that will be equals to 0 minus 1, and this will yield a value of -1 to the box. So now we will check all the three scores derived by a horizontal, vertical or global alignment. So we got three negative values when matching the scores horizontally, vertically as well as diagonally. Since all negative values are to be taken as 0, so we will fill a value of 0 within this box. So now we will fill all the rest of the boxes along to this set of rules, and you will get the scores like this. So we will see multiple set of alignments.
Now let's start the traceback here. Contrary to the Needleman-Wunsch algorithm, which states that traceback should start from the lower rightmost box, the Smith-Waterman algorithm states that the traceback should start from the box having the highest value. In this case, the highest value of 2 is observed for this box as shown here. Now from which box did we get this value? Of course, we got this value of 2 from the box having 1 in the diagonal direction. As you can see here, we have earlier studied that this diagonal direction clearly indicates a matching or mismatching. We can clearly see here that there is increase in value from 1 to 2, which indicates a matching. Moving ahead, from which box did we get this value of 1? We got the value of 1 from the box having value of 0 in the diagonal direction. Now there is also an increase in value from 0 to 1 as we go to the diagonal direction. So it also indicates a match as discussed earlier. In contrary to this Needleman-Wunsch algorithm, where traceback starts from the rightmost box and ends at the left uppermost box, the Smith-Waterman algorithm starts right at the box where we get the value of 0. So clearly this is an indication of a local sequence alignment. Now let's start the alignment. The value of 2 was observed for the nucleotide G, and this, since there is an increase in value, this is an indication of matching between the nucleotides. So here clearly G is matching with G. Further, the value of 1 was obtained for the nucleotide C, and since there is an increase in value, it means C is also matching with C. In addition to this sequence of these nucleotides, that GC, a pair of GC is matching with a pair of GC, we got two other instances of nucleotides. As you can see on your screen here, we got a value of 1 in the right side, and this value of 1 was obtained from the value of 0 in the upward diagonal direction. Fine, there is an increase in value, it means there is going to be an alignment. Of course, there is an alignment between A matching with A. Further, we got another instance of increasing values of 1 which is obtained from a diagonally positioned 0. So this is also an indication of A is matching with A. In addition to the small matching, that is A is matching with A, and here again A is matching with A, you can clearly see there is T is matching with T, and A is also matching with A at the left corner as shown in this box. So this small examples clearly explains how do we perform local sequence alignment using the Smith-Waterman algorithm. I hope now you have understood this algorithm, and or if you have any questions, then please let me know by putting a comment in the comment box. I will meet you soon in the next video. Thank you so much for watching this video.